Good morning, my dear friends. Welcome back to my YouTube channel Medical Classes by Dr. Srinidhi Kumar Acharya. Please don't forget to subscribe to my channel and also please give your valuable comments. You can also visit the playlist. In the playlist, I have provided almost all the topics in a very systematic order and you can also make use of it. My dear friends, in the last class, we are discussing about complete gonadal dysgenesis, which is also called as Swyer syndrome. Today, we will look into another important aspects of differential sexual development, that is androgen defects. So far, we have not discussed of any androgen defects. Androgen defects in the sense, the hormones which are produced from the adrenal gland. If there is any variations or abnormality or disproportion of these hormones are there, again, especially in the fetal period and the early neonatal period, if there is any problem related to the adrenal gland and secretion of the androgens, that also results in ambiguous genitalia. So today onwards we will going to see that. So far we have only discussed about the chromosomal defects. So far we have only discussed about the gonadal defects. So far we have only discussed about the improper formation of this anti-Mullerian hormone and the Sertoli cells, Leydig cells, testosterone, dihydrotestosterone and some of the enzymes, etc. But today, so we shall see actually the core portion of this DSD, that is androgen defects. Now when I say androgen defects may be the problem with the androgen synthesis. That means the androgen itself are not properly formed, or number two is androgen conversion defect. There may be problem with the conversion of the androgen into the next stage. Okay, so androgen is normally synthesized, but it is not properly get converted into its next form or next stage. So that may be also the defect. And third one is receptor defects. That means androgen receptors are not proper. Okay, that means adrenal gland receptors are not receiving any signals from the outside like testosterone and dihydrotestosterone, etc. So whatsoever may be the case, it may be receptor defect, it may be a conversion defect, or it may be synthetic defect. Whatsoever may be the case, the final presentation is always under virilization. Whenever there is no androgen, always the final result is under virilization. Secondary sexual characters are not properly developed. So that will be the presentation whenever there is androgen defects are there. Now if you take the example in androgen synthetic defect, the first one I told androgen synthetic defect. So in case of androgen synthetic defect, the 17 beta HSD problem may be there. 17 beta hydroxylase enzymes, that problem may be there. And if that is the case, if there is problem with the androgen synthesis synthetic defect, if there is 17 beta HSD problem is there, and there will be increased androstenedione and testosterone, and this leads to decrease in the ratio. So now what actually happens is, if there is androgen synthetic defect is there, 17 beta hydroxylase problem will be there, and because of this there will be increased androstenedione and testosterone will going to decrease, and the ratio will be less than 0.8. If the ratio between this androstenedione and testosterone, if it is less than 0.8, it leads to so many problems. Similarly, there is one more problem, that is 5-alpha reductase deficiency. 5-alpha reductase is related to the conversion of testosterone to DSD. We know that DHT, that is dihydrotestosterone. So if the 5-alpha reductase deficiency is there, it is not properly formed, relatively there is less formation of the DHT, but testosterone will go into increase, because DHT dihydrotestosterone is always formed from the testosterone. Now testosterone is normally properly formed, but it is not properly get converted into its next stage called as dihydrotestosterone because of the deficiency of the 5-alpha deficiency. And this results in more amount of testosterone, and this results in less amount of DHT. DHT is very important for the formation of the external genital organs, we know that. And here the ratio will be more than 10:1. Testosterone is to DHT ratio will be more than 10:1, again, which is pathological. Okay, in the first case it is between the androgen and testosterone. There will be increased formation of the androgen, and testosterone is normal. Testosterone level is normal, but compared to testosterone, the androgen level will be more. Okay, because of androgen synthetic defect by 17 beta HSD, and that leads to decreased amount of the testosterone and more amount of the androgen, and the ratio will be less than 0.8. Again, it is pathological. Now, what will be the outcome of all these problems? The problem will be the gonadal developmental problems, or there is too much of androgens. Okay, whenever this happens, this ratio get disturbed, there will be two problems because of the androgen synthetic defect problems. Okay, either there will be gonadal developmental problems will be there, or there may be too much of androgens. Androgen levels are more. The too much of the androgen is more common in the society, and it may be too much of androgens are coming, and this too much of androgens may be coming from the baby side, or it may be from the mother side. From the baby side, one of the most common cause for production of more amount of androgen is because of congenital adrenal hyperplasia. Congenital adrenal hyperplasia. Now, in the adrenal gland, we have to remember these three important things: 21 alpha OH deficiency, 11 beta 3 deficiency, and also the aromatase deficiency, that we will see in detail now. This is from the baby side. Baby side, one of the most common cause where androgen production is more, that is congenital adrenal hyperplasia. From the mother side, the contributor for production of more amount of androgen is either the mother is taking any medications, or if the mother is taking any medication for the malignancy, rather than malignancy, if there is malignancy of the adrenal gland, pheochromocytoma, it is called as pheochromocytoma. Malignancy of the adrenal gland is called as pheochromocytoma. If mother is suffering from pheochromocytoma, naturally she will produce more and more androgens, and that will be also going to the body of the baby, and it will cause the problem. Otherwise, exclusively from the baby itself, baby's adrenal gland may undergo hypertrophy, that is called as congenital adrenal hyperplasia. Both these two conditions leads to the extra amount of the androgens in the blood circulation, and that leads to the problem. It may be from the baby side, or it may be from the mother side. Now let us take the example. Now we have a baby with the ambiguous genitalia, suppose. Okay. Now first you go for karyotype. Identify what is actually the sex is. By fish type method you have to identify. And we find, yes, by after doing the karyotype, it was concluded that this particular baby is, or fetus is XX karyotype, that is female. Then you have to go for 17 DHP level. If 17 DHP level is very much increased, then it is 21 alpha OH deficiency. If 17 OH level is raised, to the moderate amount, then it is non-classical type of 21 alpha OH deficiency. And if 17 OH level is normal, then look at the AMH. AMH means anti-Mullerian hormone, because we have XX karyotype, isn't it? So there may be the gonadal problems also. But there may be some gonadal problems also. So anti-Mullerian hormone level, if it is raised, it suggests if anti-Mullerian level is raised, then it suggests that testis is there, because anti-Mullerian hormone is produced from the testis itself. Testis is there. So then it suggests that it is a ovotesticular or testicular DSD. And if the anti-Mullerian level is normal, then it may be gestational hyperandrogens, as in case of malignancy, pheochromocytoma, etc. Now, if it is XY karyotype, this is what we learned about XX. Suppose the karyotype is XX, XY, male. Then look at the Mullerian structure is present or not. If Mullerian structures are present, that means that Sertoli cells are not developed at all, because if at all Sertoli cells are there, they would have secreted anti-Mullerian hormone, and Mullerian duct would not have been developed. Okay, now in this case XY, it is, when you look into the Mullerian structure is present. That means that anti-Mullerian hormone, which has to be produced from the testis of the male, okay, is not produced. So then it leads to a gonadal developmental problems. If Mullerian structures are absent, suppose, it suggests that Sertoli cells are there and they have secreted anti-Mullerian hormone. Therefore, there is no development of the Mullerian duct and there is no female genitalia. But if the anti-Mullerian hormone level is normal, then again you have to look into the, not only the Sertoli cell, you have to look into the Leydig cells. Because Leydig cells, they will produce the testosterone. Okay, if AMH level is normal, but still child is having ambiguous genitalia, okay, anti-Mullerian hormone is not produced. That means the formation of the Mullerian structure and all that is excluded. But still ambiguous genitalia is there, no? So we need to find out the cause. So now go for the Leydig cells. Leydig cells has to produce testosterone. Now look at the testosterone level and as well as DHT level, because after testosterone there will be formation of DHT. If the ratio is normal, suppose less than 10:1, as we discussed last slide, in the last slide we have discussed, if the ratio is less than 10:1 between testosterone and dihydrotestosterone, then it look at the individual testosterone concentration. Okay, if it is 10:1, then look at the testosterone individually, specifically only the testosterone concentration. If the testosterone concentration is too much, if too much of testosterone is there, then it may be androgen insensitivity syndrome, because adrenal gland receptors are not at all receiving the testosterone. Therefore, testosterone level is going to raise in the blood and therefore testosterone is increased. If the testosterone is normal, then it may be a 17-beta HST, hydroxylase deficiency, or it may be an androgen insensitivity syndrome. If the testosterone level is decreased, that suggests that 3-beta AST problems. Okay, 3-beta AST problems. If the ratio is more than 10, okay, less than 10 we have seen, if it's more than 10, then it suggests 5-alpha reductase deficiency. I know that you are little confused here how I am telling all these things, because we have not studied. Why you are get confused here is because we have not studied the actual part yet. I am just giving the introduction. But just remember all these things, what actually happens. This is the approach. Then how 3-beta ASD problems will be there if there is decreased testosterone. Okay, if the ratio is more than 10, why I am telling that it is 5-alpha reductase deficiency. This portion I will going to explain now. Before that, few dictums will be there, we need to understand. Now we have the ambiguous genitalia and Mullerian structures are present. Suppose ambiguous genitalia is there, Mullerian structures are present. Mullerian structures are present means it is female. There is no production of the anti-Mullerian hormone. So it is definitely female, XX DSD. But we have to rule out CAH. Why? Again, I will tell you in the next class. Okay, but we have to rule out CAH, congenital adrenal hyperplasia, because even congenital hyperplasia, if there is insensitivity is there, these problems may be there. Now, no palpable gonads. Suppose there is no palpable gonads. When there is no palpable gonads, usually we will say testis is not formed, isn't it? Or ovary is not formed. Of course, ovary is not palpable. So there is no palpable gonads, plus there is no Mullerian structures. See, if there is no Mullerian structures, what you say? It should be male. But there is also no palpable gonads. Okay, then what you say about this ambiguous genitalia? So this may be agenesis or dysgenesis of the testis. Okay, it's a male actually, because there is no Mullerian structures, but there is no palpable gonads because that gonad palpability is not seen because there is actually agenesis or dysgenesis of the testis. That is also a possibility. Third dictum is palpable gonads. Gonads are palpable. Okay. No Mullerian structures are present. Yes, gonads are palpable, mostly male. No Mullerian structures are present, mostly male. But external genitalia is slightly ambiguous. External genitalia is because of the dihydrotestosterone. Okay. So, but external genitalia is slightly ambiguous. That means everything is normal. Gonad is there, antimullerian structures are not present, Mullerian structures are not present. That means testosterone is normally working. But somewhere some problem is there in conversion of this testosterone into dihydrotestosterone. Therefore, only external genitalia is absent. Fourth one is palpable gonads are there. Palpable gonads are there. And Mullerian structures are present. Palpable gonads, okay, maybe male. Mullerian structures are present. Mullerian structures are present in female. But it is present here. If you see loose testis, testis are palpable. Gonads means mostly usually will take testis. So then what is there? What may be the possibility? Then it may be an ovotesticular problem. Ovotesticular problem or AMH problem, anti-Mullerian hormone problem. In the male, anti-Mullerian hormone should be produced. If the gonads are there, testis is there, anti-Mullerian hormone would have been produced, but it is not probably produced. Therefore, Mullerian structures are present. So then this particular condition is called as ovotesticular problem. So these are few of the dictums. Now from the next class onwards, you will get total clarity about what we have discussed so far. So when we discuss congenital adrenal hyperplasia. So till then, goodbye. We will meet in the next class. Thank you very much.